Welcome to this last video where we are going to talk about non-disjunction. So what happens when something goes wrong with your DNA? And there are many different types of genetic errors leading to disorders. There's gene mutations. We're not going to be talking about those. We've been talking about chromosomes. As you can see, looking at all the pictures in the upper right, there's lots of different types of structural chromosome defects, but what I want to talk about are numerical chromosome defects when you don't have the correct number of chromosomes. So instead of having 23 pairs of chromosomes, what happens if you are missing one of the chromosomes? Or what happens if instead of having 2 of one of the chromosomes, you have 3 of one of those chromosomes? And the numerical chromosome defect we're talking about is called non-disjunction. This is what happens when chromosomes do not separate from each other and both of them end up going to a daughter cell. And so that results in an abnormal number of chromosomes. If it happens in meiosis, then you have an abnormal number of chromosomes in the gamete, where some are going to have more and some are going to lack some. It could also happen in mitosis, where it would happen to non-sex cells. It's much rarer in mitosis, and it usually ends up in the death of that cell. In any of the chromosomes, it can happen to any of the 23 pairs, including the X and the Y chromosomes. So here's an example showing what happens with the X and the Y chromosomes. This is showing a normal male where an X is in one sperm and the Y goes to the other sperm. But if non-disjunction happened with the female, where both X chromosomes went to one ovum and no X chromosome went to the other one, then at fertilization, you have a choice of 4 things. If daddy was donating the X, it fertilized mom who had 2X, you could have a triple X female. Or if it fertilized the mother cell that did not have any sex chromosome, then you would have a female known as a Turner syndrome, only having one sex chromosome, an XO female. On the other hand, if dad was contributing a Y chromosome and mom contributed 2X chromosome, then you would have a male child that was XXY. This is known as Klinefelter syndrome. But if the Y chromosome fertilized a cell from mom that did not have a sex chromosome, this is not viable because the Y chromosome has very few genes on it, and you need to have an X chromosome. As I said, most of the chromosomes are required. An absence of most of the chromosomes is going to lead to death. Okay, so non-disjunction in mitosis is going to end up with some cells which are going to be 2n+1, as in they have an extra chromosome, and some cells that are 2n-1, meaning they are missing a chromosome. They'll have 45 instead of 46 chromosomes. And this happens because when the sister chromosomes are lined up in the middle in metaphase, when they separate in anaphase, the sister chromatids do not separate. And the thing about this is not happening in our gametes. So this is happening for our somatic cells, which is responsible for growth and development. And so the cells usually die here. So non-disjunction and meiosis was where we usually see the effects of non-disjunction, because here we are making errors creating gametes. And so we're going to see the results in offspring. Also, because we have 2 divisions, we've got twice the chance to have these errors occur. If fertilized, most of these are going to cause death, especially those where you have the loss of a chromosome. Almost all of those are going to cause the death of the child, with the exception of, for instance, that Turner syndrome, the one missing the X chromosome. We do know that in the live births, if we were to do chromosome analysis of every live birth, that about 0.6% of live births do show evidence of non-disjunction. So there's quite a few of them that fly under the radar. So non-disjunction in meiosis 1 means when we have the tetrads are not separating. So we're having non-disjunction of the homologous chromosomes where all 4 would go to one cell and none would go to the other cell. So if you're only looking at, for instance, 2 chromosomes, so we've got a red set of chromosomes and a blue set of chromosomes, at the end, what you notice is that we end up with 2 gametes having an extra chromosome and 2 gametes having 1 less chromosome than normal. So all of them, if the non-disjunction happens in meiosis 1, all of the final cells will be abnormal. None of the cells will have the normal 46 chromosomes. They all will be either short or over. On the other hand, if non-disjunction happens in meiosis 2, well, in the first meiosis, the division was normal. And in the second meiosis, we're just separating out sister chromatids. And so in the first meiosis, this cell over here, well, both cells had divided normally. And in the second one, the sister chromatids are going to separate normal. And so all of these cells over here are going to be normal. And with non-disjunction happening in this cell in meiosis 2, what is going to happen is the red ones are going to split up normally, but here, both of the blue ones are going to go to one cell. And so when non-disjunction happens in meiosis 2, we end up with 2 cells that are normal, 1 cell that is missing a chromosome, and 1 cell that has an extra chromosome. And that's because in meiosis 2, the non-disjunction occurs between sister chromatids. Okay. So one of the most common non-disjunctions gives us, that is compatible with life, gives us cells that have an extra chromosome. Because as I said, most of the time when we have lost a chromosome, that ends up being non-compatible with life. And so when we have an extra chromosome, that's called a trisomy, as in we have 3 copies of it. And so one of the most common non-disjunctions occurs with chromosome 21. And the reason why this is compatible with life is because chromosome 21 is a really small chromosome. And so here is what is happening. We have a sperm that has normal 23 chromosomes, and it is fertilizing the mother cell that in meiosis had non-disjunction, and she had cells that had 22 chromosomes and one that had 24 chromosomes. So the one with 22 chromosomes died. She had cells that had 23 chromosomes. They did not fertilize, but here the sperm chose to fertilize a cell that had 23 chromosomes. So they had 24 chromosomes, and the extra chromosome was an extra copy of chromosome number 21. And so the baby has 47 chromosomes. Okay. And so every time that cell undergoes mitosis, all that baby's cells are going to have 47 chromosomes. And so if we do a karyotype of that baby's cells, what we see are 3 copies of chromosome 21. This happens to be a girl because you see an X and a Y chromosome. And trisomy 21 is Down syndrome, and this is the most common non-disjunction that is compatible with life. Many chromosome 21s end up with death early in pregnancy, where the ladies have what we call a spontaneous miscarriage um, in the first few weeks of pregnancy. But at live birth, if without any medical intervention, about 1 in 700 births would end up with trisomy 21. We know as the age goes up, the incidence of trisomy 21 goes up because the mother's oocytes are older. Okay. They develop before she's even born. And so in her 20s, they're 20 years old. In her 30s, they're 30 years old. In her 40s, they're 40 years old. And so the chance of having non-disjunction just rapidly increases the older they are up. And so prenatal testing can look for the presence of trisomy 21 by looking at cells, or even now for the presence of Down syndrome in children with ultrasounds and look for associated malformations, and patients can decide, or mothers who have children with trisomy 21 can decide whether or not they want to continue the pregnancy. And so now what we are finding is the highest age of mothers with Down syndrome is not in the 40 plus age group anymore, because many of these mothers are being scanned for and choosing not to continue those pregnancies. But actually much younger mothers are the mothers having the babies with Down syndrome, because insurance does not cover the screening for this. Okay. So as I showed before, we can have non-disjunction in sex chromosomes. And in fact, we do know that up to 5% of fertilizations do have spontaneous loss of X or Y chromosomes. And only the total loss of the X with a Y is non-viable. It is very viable to have multiple X's, only to have 2 X's, 3 X's, 4 X's with a Y. And to survive as biological females and biological males. And you won't even know that unless you have a karyotype done. Now, a lot of these people are normal, and sometimes, as in Turner syndrome, you do have a syndrome, you survive, but you are not, quote, normal. So different combinations give different syndromes, which have different clinical manifestations. So the incidence is not nearly as rare as you may think. 1 in 1,000 females have more than 2 X chromosomes. 1 in 2,000 live female births only have 1 X chromosomes, and 1 in 650 male births have 2 X chromosomes or more. So it's not a rare occurrence at all. So pull it all together, look at your concept map. Please take the time, do the studying, practice teaching it, make sure you understand it, learn the basics before you add on all the details. And I thank you so very much for putting in all the time and effort. Make sure you are putting in all the hours you need every single week. Remember, you cannot just cram for a day or two before a test in a natural science course and expect to do well. And I know you can do this. Be sure to reach out to me if you have any questions. We are here to help you. And I will see you soon.